Welcome back to Advent and to another session of Dr. Mohanty's Masterclass. Today, we will take a closer look at one of the most widely used and scientifically significant surfactants, sodium lauryl sulfate, also known as sodium dodecyl sulfate or SDS or SLS. This compound is an integral part of dissolution studies, helping us simulate real-world drug behavior and ensure analytical reproducibility. Through this session, we will explore its chemistry, its critical role in dissolution, and why the purity of this molecule can make a world of difference in analytical science. Sodium lauryl sulfate or SLS is an anionic surfactant, meaning it carries a negative charge when dissolved in water. It is known for its ability to lower surface tension, improve solubility, and disperse hydrophobic materials uniformly. In dissolution studies, SLS is widely used to enhance the solubility of poorly water-soluble drugs, ensuring that we obtain a biorelevant and reproducible dissolution profile. The molecule itself is simple yet powerful. It has a 12-carbon hydrophobic alkyl chain, the lauryl group, and the hydrophilic sulfate head attached to sodium. This dual nature makes it amphiphilic. One part loves water, the other repels it, enabling micellar formation. The molecular formula is C12H25SO4Na with a molecular weight of 288.38. It is this balance of hydrophobic and hydrophilic tendencies that makes SLS such an effective solubilizing agent. In dissolution testing, especially for BCS Class II drugs, solubility often becomes a rate limiting step. By adding a small amount of SLS, we mimic the surfactant conditions of the gastrointestinal tract. SLS improves wettability, enhances dispersion, and forms micelles that solubilize the drug molecules. This leads to improved and consistent drug release profiles, which are crucial for both formulation development and bioequivalence studies. Typically, concentrations between 0.25 to 2% are used depending on the solubility characteristics of the drug. Why purity matters? When it comes to analytical applications, purity is everything. Commercial SLS often contains residual sodium sulfate, alcohols, or unsulfonated hydrocarbons. These impurities can alter surface tension, shift the critical micellar concentration, and introduce baseline noise in HPLC or UV studies. Therefore, a high purity SLS ensures not just solubility but also analytical consistency and data integrity. The small impurity variation can mean a big difference in reproducibility. At Advent, we offer two high purity grades designed for analytical excellence. We have developed high purity SLS, 99% AR as well as HPLC grade, and an even higher 99.5% HPLC premium grade to serve both routine and high-end analytical applications. Our SLS, AR and HPLC grade, which is more than 99%, is ideal for routine QC and dissolution media preparation, and our SLS HPLC premium grade, which is more than 99.5%, is designed for high-end applications, bioequivalence, reference standards, and regulatory submissions. These grades are characterized by extremely low unsulfonated matter, controlled sodium sulfate, and minimal UV absorbing impurities. Each batch is tested to ensure consistent CMC behavior and reproducible surfactant performance. Physically, SLS appears as a white to off-white crystalline powder or flakes. It dissolves freely in water and ethanol, forming a clear, sometimes slightly viscous solution. At 1% concentration, its pH is generally lies between 7 and 9. The critical micellar concentration, which is around 8 millimolar, defines the threshold where micelles begin to form. Conductivity and surface tension measurements are often used to confirm its quality and purity. These physical characteristics make SLS both predictable and dependable in laboratory use. SLS is hygroscopic, It tends to absorb moisture from the atmosphere. If exposed to humidity or air for prolonged periods, it can gradually degrade, leading to a slight yellow or off-white tint. This is generally due to oxidation of trace organic residues. Therefore, it is best stored in airtight containers away from light and moisture. Proper storage ensures the surfactant remains chemically stable and analytically reliable. Applications beyond dissolution. Beyond dissolution studies, SLS finds use in several fields of science and industry. It is used in SDS phase for protein electrophoresis, as an emulsifier and wetting agent in formulations, and even in cleaning validation studies in pharmaceutical facilities. Its well understood micellar behavior and strong detergency make it a truly versatile analytical reagent. To conclude, sodium lauryl sulfate is far more than a surfactant. It is an analytical companion that ensures reliability, reproducibility, and scientific precision. Purity defines its performance, and performance defines the quality of our analytical outcomes. With ADVENT's high purity SLS grades, researchers can work with confidence that each result reflects the truth of science, clean, precise, and reproducible. Please contact our sales colleagues and channel partners for your requirements of this unique product and get back to us in case you have any queries in this regard. Until next time, stay tuned, stay curious, and keep exploring the wonders of the world of chemistry. Thank you very much for watching.